Hello everyone, this is Dhanashri from the Dr. Omics lab. In this topic, we are going to discuss about the understanding of VCF5. VCF stands for Variant Call Format. Actually, it's an output of Variant Calling Analysis Pipeline. As in the last part, using the GATK haplotype caller, we called the variants and that output generated, which is in a VCF file format. So it is storing the information of gene sequence variations, and it is a tab delimited text file where each line represents a genetic variant which is found in your sequence genome. Means if you remember from the last few parts, we are like from the DNA sample, we are getting the raw data. From that raw data, we like whatever the reads we are getting, we align those reads to the reference genome where we are getting the BAM and SAM file, and then we call the variation. So basically, VCF file is divided into three components. One is a meta information line, which is a multiple line prefixed by a double pound symbol or double hash symbol. Then header line, which is a single prefixed with a 1 pound symbol or a single hash, which is giving you the header, and then the data lines, which is giving you the actual information of your data. You can clearly see in the image, like the lines, these lines are nothing but your metadata lines, then your header line, and then your data line. Now, as we have actually generated the VCF file, so let's see that. I have opened the VCF file in an Excel format so that we can understand the information. And you can see here the first few lines, it is showing you the information of metadata. What information we are having? That is a description, like what is the file format, then what filtered GATK applied by default. It's a low quality filter, means whatever the variants we are having with the Fred score less than 30, it is going to discard automatically. Then format, like what information we are having in a format column, that description you will be able to see here. Then in info column, so this is what a description is provided over here. Then you're having here the header line and then the data line. So let's see now what information is provided in each and every column. So the first column is a chromosome. So basically it's a chromosome name or identifier where the variant is located. Second, we are having that is a position, which is a position of your variant, like where you're that actually the position of variant on the chromosome. So as we just use the 17th chromosome, so that position on 17th chromosome it is showing. Then ID, if any unique identifier is provided, if it is available, you will be able to see maybe any uh, database ID, any mutation ID. But right now in our data, we don't have that, so it is empty. Next column we are having that is a reference, that is reference allele at the variant position. Then alternate allele, which is alternate allele observed at the variant position. Then here you are having that is a quality score, which is representing the confidence in the variant call. Then filter, like a filter status, which is indicating whether the variant passed certain filtering criteria or not. Like the filter column in the VCF file, it provides the information about the quality of variant call and whether it has passed a certain quality control filters. Means it helps researcher to access the reliability of the variant call and decides which variants to include or exclude from the downstream analysis. Means if you are getting here the pass as a filter column, if you are in any particular row, if you are getting a pass, so if the value in the filter is pass, it is indicating the variant has passed all the quality control filters which is applied during the variant calling. But as we didn't apply any specific filter, so we are not able to see any information over here. Then in info column, so basically this info field in the VCF file, it is providing the additional information about each variant, like it contains the various key value pairs, which is describing the attributes of the variant, such as allele frequency, depth of coverage, functional annotation, and many more. So, if you are not aware about any one of the term, you can read it in the metadata line. Now, the last column we are having, which is a format column and one of the most important column. So basically this format field, it describes the format of the genotype data of each sample which is including in a VCF file. Right now, as we are just working with the one sample, so for one sample it is showing, but maybe if you have a multiple samples, so call this samples are presented or they are represent in the columns and variants they are representing in the row. So this format fields it describes the genotype. It specifies the structure of genotype information, including genotype calls, read depth, genotype quality, and many more. Now, if you will see this format, basically this format is representing these values. Just to understand this better, I will just copy this format and I will paste it into the paint. Now, after this, I will paste this value because this format is representing these values over here. Now, basically this format is separated by the colon. So the first value here we are having, that is a genotype. So genotype, it suffer the represent the genotype of a sample at variant locus. Means these genotypes are typically represented using a combination of digits separated by the slashes or pipe symbol. Means this genotype, it will tell you whether your variant is a homozygous or a heterozygous. Homozygous means what? Like a same allele are coming from both the parent, like either dominant dominant or recessive recessive. And heterozygous means what? One dominant, one recessive. Now, here, if you are getting the numbers like 1 by 1, or you are getting like a 0 by 1, yeah, sorry, if you are getting a 1 by 1 or if you are getting a 0 by 0, so at that time you can say this is a homozygous. But maybe if you are receiving like 0 by 1, so at that time you can say it is a heterozygous. Where this 0 is representing the allele in the reference and 1 is your altered allele. Means, let us consider this is what your reference genome, where your reads are aligning, and here in the reference A is present, and let's consider at this position you are having total 10 reads. Out of that, 5 are supporting to the A, and next 5 they are supporting some another allele. At that time, how it is going to represent in the 0 by 1 format? So 0, because this is supporting to the reference allele and this is your altered allele. So it will be 0 by 1. So this is how basically it is going to represent. And depend based on this, there are three types of homozygous and heterozygous variants are, like if you are having a 0 by 0, if the genotype is 0 by 0, so it is a homozygous variant but for reference allele. Then if you are having a 0 by 1 as a combination, as a genotype, this is a heterozygous variant. And if you are having a 1 by 1 as a combination or as a genotype, this is a homozygous variant but for altered allele. So this is how, depend upon or based on the 0 and 1, we can identify the genotype. Next value we are having over here is for the AD. AD is nothing but the allelic depth. So here the AD they are representing as 0, 4. Basically, this allelic depth, it is providing the breakdown of total depth into the number of reads supporting to each allele at the variant locus. Means, for example, if you are getting AD for this uh, variant, we are getting AD as a 0, 4, indicating that 0 reads are supporting to the reference allele and 4 reads are supporting to your alternate allele. Okay, so this is what a allelic depth. Now dp. So dp is nothing but a depth. So here we are getting the depth 4. So dp indicating the depth of coverage for variant locus in the sample. So it is representing the total number of your sequencing reads that align to the particular position. Now we are having the genotype quality. So basically genotype quality, it is estimate the likelihood of genotype that calls incorrect. Means it is also a Fred scale where the higher score indicating higher genotype call, higher the confidence. So this is what a genotype quality. And the last one we are having, that is a Fred likelihood PL. So PL, it is represents the Fred scale likelihood of a different genotype calls. Means those scores are logarithmically transformed probabilities, which is indicating the confidence in each possible genotype. Like a lower PL suggests a higher confidence in the genotype. And here we are getting a three formats, like three values of the Fred likelihood. We are getting here the three values of the Fred likelihood, which is indicating your three types of variant. These three types of variant. Like the first is a homozygous for a reference allele. Then next you are having that is a heterozygous, and the third you are having that is a homozygous alternate. Now this value, like lower the value, higher the confidence we can say. So if you will see for the third position, which is homozygous alternate allele, this is showing you the very lowest value 0, and you can see this is also the same thing it is representing. It is a homozygous but for the alternate allele variant. So we can say that lower the value, higher the confidence of a genotype. And this is how we can predict the result for the format. We can predict the VCF file, variance in the VCF file. Now you can see at a specific position, it is showing some insertion deletions as well. So there are so many variants we are getting over here. But for those variants, we are just having the information of positions, genotype, but we are not having the information about the gene, its impact, then whether it is known or novel variant, whether it is from the coding or non-coding region, what is the consequences of it, whether it is changing the protein or not. We don't have that information, and for that we need to perform the annotation. And that annotation we are going to perform in a variant effect predictor, which is the online tool we are going to use for the annotation, and that we are going to discuss in our next part. If you have any question here, please let us know. Till then, bye. Have a great day.